Good evening and welcome to the penultimate day of the faculty development program on next gen AI training. We have been going through the various basic ideas of modern AI, from the basic machine learning algorithms to large language models. And now it is high time to explore some latest research areas in modern artificial intelligence, and the remaining sessions will give insights into these kind of areas. And today we have our resource person Akshatha Nayak. She is the first overseas resource person for this faculty development program. To introduce her and to welcome her into this faculty development program, let me invite our beloved HOD, Dr. Unnikrishnan K. Thank you.
Thank you, sir. Actually, we are really proud and happy to introduce our dearest student, Akshatha, to this faculty development program. Really, I am fortunate to invite my students, our department student, to this faculty development program. Actually, while studying, she was a role model for the entire department. We were sure that you will reach very heights in your professional life. And you have reached at this stage, surely you will go or you will reach really very good heights again. Okay, let me introduce formally Akshatha. Akshatha is a bioinformatics researcher specializing in computational cancer genomics. Her work focuses on applying statistics, data science, and machine learning to study the disease. She previously worked as a bioinformatician at the European Bioinformatics Institute in UK and is currently pursuing a PhD in computer science at the University of Texas at Austin. Thank you, Akshatha, for agreeing to take a session for our faculty development program in spite of your busy research schedule. Thank you very much. You can start.
Thank you, Unikrishnan sir, for the kind introduction. Am I audible? Can you confirm?
Yes, yes, you are.
Yeah. So thank you very much for the introduction. I'm very honored to be invited for the talk here, especially because Unikrishnan sir was my class advisor while I was at RIT. So I'm really happy to be back now to give this talk. Let me share my screen.
Maybe actually your mic is mute.
Can you hear me now?
Yes, yes. Now it's
Okay.
audible.
Okay. Just give me a minute. Can you see my screen now?
No.
Do you see my slideshow?
No, it was visible a while before, but now it's not.
Sorry?
Now it's not visible. It is not.
Previously it was visible. Now it's not visible. Try to present again. Now it is visible, but you have muted your mic.
Okay. Okay, I think,
Ah,
and is show also visible?
yes, yes.
Okay, great. Yeah, sorry for the network issues. So, yeah, today I'm going to talk about applications of machine learning in bioinformatics. And bioinformatics is an area I'm really passionate about. So I'm very excited to talk about the applications of ML in this area. The agenda for today is going to look like this. I'll first introduce briefly the area of bioinformatics and what the applications of ML are in this area. So I'll here talk about how traditionally machine learning has been applied. And then I'll fast forward to what the current newer methods are for applications of AI in this area. So there are two big challenges that have been solved recently. One is the long-standing challenge of protein folding. So this challenge was solved using this model called AlphaFold. I'm sure some of you may have heard of it because this was a Nobel Prize winning model last year in 2024. I think it won the Nobel Prize for chemistry. And then the next big leap happened very recently, where they tackled the dark matter in our DNA. So this is the part of DNA that is not very well understood through conventional studies. So this was solved using Alpha Genome, and it was released very recently. And after I talk about these two AI models, I will go ahead and talk about what the future direction looks like for the field of bioinformatics, especially pertaining to machine learning applications, what the challenges are, and what the exciting things are to look ahead. So just to give a brief intro to bioinformatics, I'm sure many of you might already know this, but bioinformatics is the area of intersection between computer science, biology, and statistics. So this is sort of like applying data science to biology. And there are many different kind of things that are incorporated in bioinformatics. So for example, it could be building different tools that help researchers solve biological problems, or it could involve developing novel methods and computational algorithms that help process biological data. It could involve building different kinds of databases. So it could be databases related to DNA, databases related to clinical data of patients, etc. And then analysis of this data to derive insights and also to answer biological questions. Finally, it also involves performing simulations on biological data to model different kinds of biological systems and processes. So to talk a little bit about the different kinds of data that we deal with, the most common one we may have come across is DNA, RNA, and protein sequence data, and then the different types of data that we derive from this. For example, assemblies of genome sequences and variant data. So variant data is, you might know, our DNA is pretty similar across different humans. I think 99.9% of the DNA is similar, but there's this 0.1% difference that we have. And these differences are in the form of mutations in the DNA. So we also study these mutations, and they form a very important part of biomedical studies within bioinformatics. So that's one kind of data that we look at. We also look at network data. So people study protein interaction, so different proteins and how they interact with each other. And to study this, we can use graph data structures and different graph algorithms to model these. We also have structural data. So I just talked about the problem of protein folding and protein structure. So this is related to that. Here, we look at how the different units within a protein fold and associate themselves with each other to form a 3D structure. And this data is in the form of differences between different units and different angles at which they're concorded. So that's structural data. We also have clinical data. So within this, we have text data, which is electronic health records. These are health data that doctors record when we visit them. We also have pathology reports, which are in the form of text. When it comes to image, we have radiology scans and data that we get from microscopy images. We have numerical data such as lab test results and vital signs like blood pressure, pulse, and time series data. So what's time series data? It's so consider an example where a person has been going to the doctor every month or like every other month for the last 10 years. So this happens when you have a long-term disease like blood pressure, and that needs to be monitored frequently. So on every visit, certain vital signs are recorded. And if you look at the data for over 10 years, you can see the progression of how the vital signs have changed over the last 10 years. So this kind of data is really important for research because it helps us understand what kind of medicines may have led to improvement of disease over time, and how different patients are responding to different diseases. So this is also an important kind of data that we look at. And this kind of data is also one which can be analyzed really well with deep learning models, such as we have LSTM, which looks at time series data. So those can be applied for electronic health records, which are in a time series format. Uh, and also, let me give you some examples of applications of traditional machine learning model and biology. So these have been around for a while, and I would like to start with this. First is predicting protein structure. Earlier attempts had been made to do this with traditional models, but they were not successful. So today I'll talk about what the current state of this area is. We also have predicting effects of mutations. So I just mentioned a while ago that we have about 0.1% of difference among us when it comes to the DNA. And these differences lead to the visible differences that we see. For example, these variations determine what our eye color would be, what our hair color and skin color would be, our height, sometimes even our tendency to gain or lose weight, uh, our predisposition to different diseases, and those kinds of things. So if we can look at a mutation in the DNA and predict what its effect is going to be like, that would have tremendous benefits for different kinds of research. And that's also related to the next point, which is predicting disease risk. So if we can understand the effects of mutations, they would also help us more accurately predict the risk of different diseases. So all of these are examples of how prediction-based methods are used in bioinformatics. We also use classification methods. So this might be a very common example you're familiar with. We can use methods to classify, for example, tumor versus normal tissues. So we will have images that we get from different kinds of scans, and machine learning models are able to better identify or segment parts of these scans which indicate that there is a tumor. So when these kinds of models started out, even if they performed as good as a doctor, that would be huge. But recently, there are models which can even predict uh, tumor better than doctors. So even if there is a very small tumor, which a doctor might miss when they look at the image, these methods have been able to identify them very accurately. So this is another area which is making tremendous leaps. And then another example of classification in this area is classifying patients by drug response. So we have different kinds of drugs for different diseases, but people respond differently to each of them. So for blood pressure, we might have 3 different kinds of drugs, but how I respond to that is going to be different from how some other person responds to that. And so for a given drug, we can classify if you will respond well, like which population is going to respond well to the drug and which population is not. So this will help doctors determine what kind of drug to administer to a given patient. So this is a very important problem. And finally, we have clustering based machine learning methods in this area. So some examples of application include sequence clustering by similarity. So here, these are used to determine, to understand evolution. So we look at DNA sequences of humans along with other organisms, and we see how similar they are. And the more similar they are, the more recently evolved they are. So that way we can sort of form an evolutionary tree using sequence clustering. And this is also used for identifying disease subtypes. So for example, let's consider a population of patients who have lung cancer. Now, even though everyone has lung cancer, the nature of the cancer is going to be for different for each of them. And depending on the type of cancer, we can group them into different subtypes. So for example, let's say for lung cancer, we have type A, B, C, and D. We can look at different symptoms that the patients are presenting, different results that we get from scans and tests, and then we can identify what specific type of lung cancer they have. And the reason why this is important is because this helps us determine the treatment course for the patient. So if the patient has lung cancer type A, they would be given a particular medicine. Some other patient might have type B, which might be more severe, and they don't respond to this first medicine, so they have to be given some different medicine. And so being able to subtype the disease is very important in a clinical setting. So this was an example of clustering. Now that I've talked about the traditional models and their applications in different areas of bioinformatics, mostly focusing on biomedical research, I'll now talk about protein folding, which has been a long-standing challenge. So protein folding refers to the process by which the protein sequence assumes its functional shape. So if you look at the figure here, let's assume this is a one-dimensional representation of the protein. So a protein is formed of units called amino acids, and it's a sequence of these amino acids that makes a protein. So this sequence can then turn into helical shapes or sheet-like shapes, and that kind of formation is called the secondary structure. And further, this formation folds upon itself, or like forms crevices and bonds, and it forms a 3D kind of structure as shown here, and that's called the tertiary structure. Finally, different protein units come together and form a functional unit, and that's called a quaternary structure. So this protein folding problem refers to how we go from this primary sequence, which is a one-dimensional sequence, to this tertiary structure, which is a three-dimensional sequence. And that is what we are going to look at in this section. So why is this important? This helps us understand protein functioning, because when you look at tertiary structure, you see there are these different gaps and different atoms that are exposed, and there are some atoms which are not exposed and hidden instead. So this lets us understand how this protein is going to interact with other proteins and what the function of this protein could be in our body. So this helps us understand protein functioning, in short. And this also helps us understand the impact of these changes on diseases. So different kinds of variations in the DNA sequence can translate into different structure of a protein. And many diseases that we see are actually related to different protein structures. So these changes can, being able to understand how proteins fold and how variations lead to different kind of folding, can also help us understand its impact on diseases. Third, it helps us in drug discovery and design. So the way medicines work is they interact with proteins and either inhibit their function or increase their function or do different kinds of things. And that's how medicines solve different symptoms. So what we can do is we can use this protein folding problem to discover new drugs, because drugs themselves are also structures like this. So if you're able to predict structures, you will be able to predict at least like a set of different drugs that can potentially do the kind of thing that we want it to do. So if this protein folding prediction algorithm is not there, we'll have to test out each drug in an experimental setting, and that is very time consuming, which brings me to this next point. Conventionally, protein folding is done, like predicted by looking at X-ray crystallography methods, cryo-EM, and NMR methods. And all of these are costly and very time-consuming, as all experimental methods are. So if you're able to predict this, it saves a lot of time and a lot of money, and it helps in accelerating research. So the model that solves this problem is AlphaFold. This was a model that was discovered by Google DeepMind in 2018. So in 2018, they came up with, I think, the initial release of the model, and since then they have made improvements. Currently, they are on AlphaFold 3, but I'm going to talk about the initial model that they released as a paper where they have the structure. So that's, I think, AlphaFold 2. So to put it simply, we go from the input sequence, which is the sequence of amino acids. So just to brush up your memory, this is what it looks like. So we go from this sequence to this 3D structure over here. That is what this model does. And because this is complex, I'm going to break this down in the next few slides and talk about each part of the model. So first, I'll talk about this part. So this part refers to MSA representation. MSA means multiple sequence alignment. What this does is you have the amino acid sequence from the primary organism. So for example, if you're looking at human protein, that would be human. And then we also have the sequence of the same protein from other organisms. So for example, we know hemoglobin is a common protein we might know about, and hemoglobin is actually formed of 4 different subunits of protein. So let's take one of the subunits. We have the sequence for that subunit here, and then we'll look at the same subunit of that protein in other organisms. So in fish, in rabbit, chicken, we take the same protein sequence. Now, since this is the same protein and does the same function in different organisms, we expect that it will have a similar structure, but it does not always have the exact same sequence. It's close enough, but it's not exactly the same. So that's why we can't do a string matching algorithm. Instead, we have to do alignment of different sequences to find the ones that are similar. So we use a genetic database that has all protein sequences and find similar sequences from other organisms. That's step one. So this is one way we transform this input sequence. The next, I'll also go into a bit more detail about why this is relevant. So if this is the protein sequence from, let's say, human, these are the sequences from different organisms. So each row corresponds to one organism. As you can see here, most of these units or amino acids are going to be same, but there are some positions where they vary. For example, at position 3 and position 5 from the end, we see variations across different organisms. So when the amino acid is the same across different organisms, we say that it is being conserved. And then there are positions which are more likely to mutate, where you see these differences. So when there are mutations, because the end structure is going to be the same for each of these organisms, or at least sort of similar, we can't have random mutations. So if there is a mutation here where it changes from R to K, we have to make sure that that is another mutation which is compatible with this change. Because let's assume this is R and this is D, they form sort of a bond. And if you're changing this R, then this D is not going to interact with R in the same way, and that will mess up the protein. So if R changes to K, you also have to change D to another amino acid which is compatible and forms the same kind of bond. So in this way, mutations happen not just in pairs, but in some sort of a pattern, and they are said to co-evolve. So different positions have changes that evolve together. And this is the kind of knowledge that we need the machine learning model to train on, because that will really help it in predicting the final structure. So we looked at this part, which is the MSA representation. Now we'll look at this part, which is the pair representation. And this part helps us in learning the different interactions between different amino acids. So for pair representation, what it does is it encodes the relationship between these different amino acids. And we get this pair representation from templates. And templates are actually the 3D structures of protein that have the same that are known to have the same structure as a protein of interest. So this sort of helps as a reference for the model to learn from. And pair representation is in the form of a matrix like this. And what it encodes is actually this part here. So if we know that within a sequence, the part I interacts with part J. So the way the matrix is generated is we have the sequence here along the columns and also along the rows. So if the second position interacts with the fourth position and fourth position interacts with the sixth position, then we know that second position is somewhat close to the sixth position. So we sort of represent this as triangles and try to predict or understand the interactions in relation to each other. So this pair representation helps us model interactions within the sequence. Now we have two different representations that we obtain from the input sequence, and these representations are going to be the input to evoformer block. Evoformer is one of the core components of this model, and it's this is the transformer part of the model. So this is what the evoformer block looks like in detail. So for this particular block here, we zoom in and this is what we see. So in the evoformer block, we have two different transformers that are working together. And so it's called a two-tower architecture. We have one tower which deals with the MSA representation, like learning from the MSA representation and refining that. And then another tower which refines the pair representation of the model. The MSA representation part helps the model learn the sequence variations, and the pair representation part helps the model learn the interaction between different amino acids. So if we go to the MSA transformer part, there are two kinds of attention. I think you might be familiar with attention. It's what's also used in LLM. It helps us identify which parts of input are relevant to predicting the output and how relevant they are. So in MSA transformer, we have row-wise and column-wise attention. So this is the row-wise attention part, and this is the column-wise attention part. In the row-wise attention, it identifies which pairs of amino acid are more related to each other. And in the column-wise attention, it determines which sequences are more informative. So if you remember the MSA that we looked at, columns are different sequences that come from different organisms. So in column-wise attention, we determine which of these is more important to the final outcome. So that's the MSA transformer part, which is the top part over here. And next is the pair representation transformer part. This captures which amino acids are likely to interact with each other. And here the kind of attention that's used is called triangular attention. And this is based on the mathematical theorem of triangle inequality. So that states that the sum of two sides of a triangle is always going to be greater than or equal to the third side. So that is the knowledge that they use here to do triangular attention. And how we use it here is if we know the distance between amino acid A and B, and we know the distance between amino acid B and C, then we can set a constraint on the distance between amino acid A and C. And in that way, we are able to understand the interactions between different pairs of amino acids. So that is this part over here. And these are the triangle updates that are done as part of this model using the triangular attention mechanism. And other than having these two transformers, they also talk to each other and exchange information. And based on this exchange information, the MSA and the pair representation are updated. And this exchange and iterative update happens 48 times throughout the model. And after that happens, we have the intermediate MSA representation and the pair representation. So we covered the evoformer part, and we have this MSA and pair representation. And now we look at the next most important and the final module, which is the structure module, which generates this final 3D structure. So before we go into the details of structure module, let's talk about how these amino acids are represented mathematically. So in chemistry, these can be modeled as gas residue, and gas residue is basically it can be represented as this triangle of 3 different atoms which form the backbone of the amino acid. And what the AI model does is it moves these triangles around, so it rotates them, changes the distance, changes the distance of one particular amino acid with respect to another. So these kinds of transformations are applied to learn their final positions, and these transformations are encoded as a matrix, as shown here. So this 3 into 3 matrix represents the rotations that are applied to this, and this matrix over here represents the translations, that is movement along a particular axis that is applied to this. And this is initialized by setting all the rotation to identity and all the positions or the translations to origin. So it starts from 0, 0, 0. So that representation is what we have over here. We have a single representation for each of the amino acids that form the protein, and that is the input to this block that you see here, which is the structure module. Along with that, we also take as input the pair representation. So if we go back here, we had the pair representation that we got from evoformer, and we had the MSA representation from which we take a single one as input to this block. So these two are going to be the input, and the single representation is in the form of a backbone of 3 atoms, which I showed here in the previous slide. And the final outcome is the 3D structure. There are 2 things that I want to highlight about the structure module. First is invariant point attention, or IPA, that is this part. What it indicates is that translations and rotations don't change anything about a particular amino acid. So for example, if you have this amino acid and they are at a particular angle and distance from each other, the different atoms in it. So if you rotate the whole thing, it's still going to be the same amino acid. If you move it from one place to another, it's still going to be the same amino acid. So this IPA module is based on that concept. And another interesting thing that the structure module does is, instead of treating these amino acids as sequence, which is how they are biologically, it treats it as a bag of amino acids. So if you have a sequence A, B, C, D, for example, it will not care that B comes after A and C comes after B. Instead, it will just look at it as a collection. So in computational terms, you can think of it as a set, for example. So how this helps is it lets the model break these down and move the amino acids around. And that helps the model learn by avoiding certain constraints there. And the final structure we get shows the rotation and translation of each of these individual units and their relative position to each other. And then we have the final 3D model. Now, another thing to talk about here is the loss function. There are many loss functions here that are used throughout the model, but I'll focus on the last one, which is frame aligned point error. So this is the final loss function, and it compares the predicted atomic positions to its true positions under different alignments. So you can think of this gray scribble here as the original structure, and the green scribble here as the predicted structure. So at each position, it will look at how far away it is in distance from the predicted structure, and it will also look at the angle. So if between 2 amino acids you have a certain angle, but the predicted structure has a different angle, we take that difference as well. So those concepts are used to model this frame aligned point error. And now that we have looked at the architecture, we have looked at the loss function, let's talk about how this is trained. So For training, the data that we use is PDB. PDB is Protein Data Bank. It is a database that is maintained by the European Bioinformatics Institute, EMBL-EBI, and they have they have protein structures for a lot of different proteins, and these are structures that were obtained by experimental work. So I talked about how we have X-ray crystallography and NMR. So these are methods that are used to look at the structure of a protein, and that's the real structure that we see from experiments. And this is the data that we use to train the model. There's also another interesting thing that they do here. They use the concept of self-distillation. So here, what they do is they have a teacher model and a student model, and the student model trains on information from the teacher model by adding some, doing some data augmentation and adding a little noise. To do that, in this particular case, what they have done is they use traditional supervised learning using PDB or protein data bank data. And using this, they predict the structure of 350,000 different sequences that they obtain from this database called UniClust. And then for these, then from this predicted sequences, they obtain a subset of high confidence sequences, and then they take these high confidence sequences and put them back with the original PDB data. So the final training is done on a set of original PDB data and a subset of these predicted structures. So in this way, they are able to increase the accuracy of the model. And the kind of data augmentations that they perform here include cropping. So if there is a sequence of length 100, they take a slice of that sequence, maybe of length 80. And then another kind of augmentation applied here is subsampling. So in MSA subsampling, if we go back here, this was the MSA, and these are the different sequences that we aligned to form the MSA. You take out only some sequences for the next step and remove the others, and that's how they do subsampling. Yeah. So using this, they're able to improve the final accuracy of the model. So to summarize, AlphaFold is a model developed by Google DeepMind. It was released in 2018, and it predicts 3D structures of the final protein from the amino acid sequences. The input to this model is the primary sequence, which is the one-dimensional amino acid sequence. In computational terms, you can think of it as a string. And then from this, we derive the MSA, which captures the evolutionary information. And when possible, we also get templates for the protein structure. And templates are proteins from other organisms that have the same structure. So we use that structure as a starting point for our further steps. Then for training, we train it on the protein data bank, PDB data, and we also include high confidence predicted structures from the teacher model. And for the loss function, we use FAPE, which combines distance prediction and also torsion angle accuracy. And finally, after all of this, we have the 3D structure of protein. We now talked about what the architecture is like and the computational details, but I would also want to talk about the impact it has made. So there's this competition called CASP that has been held for a really long time, and conventionally in this competition, different people would come together and use various methods to predict the structure of protein, and whoever comes close would be the winner of the competition. When after this competition started, every year there would be improvement in performance as people came up with different algorithms to do this. People also crowdsource support from different people to get more computational power and those kinds of things. So in these competitions, I think they reached about 40, 50%, maybe 60% accuracy, and that plateaued. Once they reached there, for several years, the performance didn't increase, and sometimes it even decreased. So that was the limit of what we could do with conventional methods, with basic mathematics and basic computational science. But then AlphaFold released this model, and they participated in this competition. and they were able to get near experimental precision. That means if you do these different experiments like X-ray crystallography and NMR to get the final structure, and then you look at the result from AlphaFold, it touched the 90% mark, which was considered amazing. So it got breakthrough accuracy through this model. And another great thing they did was they predicted the structures for most of the known proteins and made it freely available through this database called AlphaFold DB. It is an open database that is hosted by EMBL-EBI. So they collaborated with DeepMind to provide the infrastructure and hosting for this huge amount of data as part of their protein data bank. So now if you go to protein data bank, you can actually see the predicted structures of AlphaFold, predicted structures by AlphaFold there for each of the proteins. So what this means is that now if you want to do some research on a particular protein, you don't have to sit and wait to do all the experiments and all the hard work to get the structure. You can actually use the structure predicted by AlphaFold because it is pretty accurate. So, and every year just gets better. So this is a great global resource for research. And yeah, so in this way, it dramatically speeds up drug development. I mentioned earlier how protein structure is related to drug development. So not only drug development, it also speeds up research in enzyme engineering and understanding the functional effects of different proteins and their interactions. And for the mark that they made in this field, it was awarded the 2024 Nobel Prize in Chemistry because it has completely revolutionized this field. So here we see how AI has changed the course of a particular field. And now I'll talk about one of the most recent AI models in this area, which is used for tackling the dark matter in our DNA. So the dark matter in our DNA refers to the parts of DNA whose functioning we do not understand really well, and AI has been able to help us better understand the functioning of these parts. So as we know, DNA contains the blueprint for the biological function, and the regulatory code for it is very complex and context dependent. So not only do we need to look at the specific parts of DNA, we also need to see how different parts of DNA interact with each other. And this makes things very complicated. So a change that happens, for example, in this part of the DNA over here might also affect this particular part of the DNA because of how it's folding over each other. So this makes predictions from DNA sequence very complex. And these modern deep learning models, LLMs, these are trained on the English language grammar or even the grammar of other languages. We can use a similar approach in in case of DNA by treating DNA as a language. So even in case of DNA, we have specific context. For example, in the English language, we have 26 letters. In case of DNA, we have 4 letters, A, T, G, and C. And then in the English language, we have a set of grammar, we have verbs, we have nouns, and how they should form a sentence structure. That's the case in DNA as well. We have different units of DNA called genes, and each of these units have subunits, and they have their own function. And there are biological constraints on how they interact and uh, what kind of letters can form those, for example. So if we treat DNA like language, then we can use LLM kind of models on DNA and learn patterns from it. So that has been the motivation for a lot of these new models. And using that concept, we have been able to solve this prediction from DNA sequence problem. And since we treat it like a language, we can use transformer architecture. And the current transformer architectures enable us to model really long contexts, like 100 kilobases or long. So that really helps us accurately do functional prediction. Why long context is important is because, like I mentioned here, a change that happens in the DNA in one place may be related to another part of DNA, for example, here. And as you can see, if you look at the sequence as a one-dimensional structure, it's very far away. You have to go all the way over here. But since DNA is wrapped around within a cell, this sometimes is located very closely to this and they interact with each other. So we should be able to model as long context as possible, and the current architectures model about 100 kilobases of this context. So, so there are some examples of different models that have been able to do this. One of the first ones which gained popularity, sorry, just a minute. So one of the first models that gained popularity was this informer model. Informer model did predictions from DNA sequence, but it was limited to understanding its functional effect only in a specific area. Then came Borzoi, which was also developed by the same organization. So Informer was by Google DeepMind, and Borzoi was a collaboration between Google DeepMind and Calico, which is also an Alphabet company. So they improved upon Informer. And then came AlphaMisSense. AlphaMisSense was able to predict the effects of different variations within DNA sequence. So that was a great leap. And what we have finally now is Alpha Genome, which was released very recently. And we'll talk about Alpha Genome now. So this was developed by Google DeepMind, released just this year. In fact, it's not fully openly released yet. They have like an overview of the architecture, but they have not released their models and their weights. So it's still very new. At least last I checked, that was the status of the model. And what Alpha Genome does, it predicts how the mutations in DNA sequence impact a wide range of biological processes. So you have a DNA sequence like this. Using Alpha Genome, it will show you what this DNA sequence would do in terms of different molecular characteristics or biological processes. So these are not relevant to go into detail, but like one of the characteristics is gene expression. So this is how much product a particular part of DNA would produce. That is DNA accessibility. So how much different regions of the DNA are exposed. Knowing that, it helps us understand how much they would function. And all of these are different molecular characteristics when it comes to genomics. And what Alpha Genome does is, based on the sequence that you provided, it gives us a map of what it would look like for all of these different molecular characteristics. Traditionally, we get these maps by doing experiments. So for each of these modalities, we have to do a separate experiment. And each of these experiments can take really long, sometimes days. So even to understand one, like for example, if you have the sequence and you change this G to C, even to understand the effect of that change, you'll have to do a bunch of experiments, look at these different maps that are generated from these experiments, and then compare them with the maps that we get from the original DNA sequence, and then do analysis. So that's a long step. But what Alpha Genome is able to do is it directly predicts in a single API call. It can give the predictions caused by that one variant change, and it gives all these predicted maps. So this really speeds things up. But of course, because this is a new area that AI has ventured into, we can't be sure about how accurate they are. And when it comes to real experiments and real research, we can't rely on this for our insights. But it definitely helps speed things up, because it helps shortlist what variations we should look at to do further experimental research and those kinds of things. So let's dive into the model architecture now. Like I said, they have not released a detailed model architecture. So we'll look at the overview that they have shared. And to summarize, the model architecture comprises of 5 different parts. The first step is convolutional encoder. Okay, let's start with the input actually. So the input is actually a 1 megabase DNA sequence. That's really long. The current models that we had was not able to do it for such a long sequence. So that was a big leap. Then this sequence is encoded. So for that, we use one hot encoding. And one hot encoding is a common concept in machine learning, so might be familiar with it. That encoding is further downsampled sort of. So we go from 1 megabase to a shorter or like a smaller representation using convolutional encoding. That's the first step. And then, so convolutional encoders, they help capture the local sequence motifs. Motifs are different patterns in the DNA. So they can capture these local patterns and short range patterns, and sometimes even long range patterns. That would be the first step of the model. The next is we have these transformer blocks. We have not one, but many of these transformers. And these transformers are interconnected through pairwise interaction modules. So these are used to exchange information between different parts of DNA. So for example, we have this part of the DNA represented by this line, and the encoder then compresses it into a smaller representation, and that goes into the transformer. Similarly, we do that for all of these different parts, and we have all of these different transformers that take as input these encoded information. And because we want to study the long range context, for example, I mentioned before that a change in this part of the DNA can also affect this part of the DNA. So to be able to study that, we have to exchange information between these transformers and understand their interaction. So that's what pairwise interaction modules will do. Finally, we go from this smaller representation back to the original size, which is the 1 megabase length. And that is done by decoder. So it goes from the smaller representation back to the original size of the representation. And after this, the final step is task-specific output heads. So some models give a single output. For example, if you have a classification model, binary classification model, it will give you 0 or 1, where 1 will be one class and 0 will be another class. That's just one output. But this model gives you multiple outputs, and each of these outputs are predicted using separate modalities. So, for example, I showed here that we have different biological characteristics. For example, we have gene expression, DNA accessibility, histone modifications, and all of these biological characteristics. And these are the experiments that are done to in real life. We do these experiments to get these particular maps, as I had mentioned. So what this model does is it predicts the maps for each of these experiments, and it does it for all of these different kinds of experiments in a single go. So just from a single sequence, it gives you all of these. And yeah, so that when you look at all of these together, you're able to understand the functioning of DNA in a lot of detail, almost equivalent to experimental. But to be able to trust this fully would take some time. As other models also do this, we can compare the predictions from one model to another, and then be able to gain more confidence in the output that we are seeing. So to summarize this model, we have a 1 megabase raw DNA sequence with single base resolution. So that means the input we have each of these individual individual characters like A, G, T, C, and all of their positions. So we have it in single base. We do one hot encoding and embedding for that sequence. And the next step is the hybrid encoder-decoder backbone, which I showed here. So this part forms the encoder-decoder backbone. That involves the convolutional encoders, which capture the local motifs and short-range patterns. We have the transformer layer, which is these, along with the pairwise interaction modules, which look at the interaction. So this helps model long-range dependencies and interactions across the input. And finally, we have a unit-style decoder, so that restores the base level. That restores the base level representation from this compressed representation. And at the end of the model, we have multimodal prediction heads. These output thousands of genomic readouts across different modalities, as I talked about: gene expression, chromatin accessibility, splicing, transcription factor, etc. And another interesting point here, which I forgot to mention, is that it does this not only for humans, but also for some other organisms. Right now, they have started with human and mouse, but as they get more data, it can easily be extended to different organisms. So that way, we can not only predict the functional effects in a human DNA, but also in mouse DNA, and maybe in the future, other important organisms that we use for research. And another interesting thing is in biology, the same DNA sequence can have different functions and different effects depending on where it is in the body. So in the skin cells, it has different effects. In the eye, it has different effects. So that is how we have these different organs, right? Because we have the same DNA, but it does different kinds of functions in different parts of the body. And that is an important thing to be able to predict for research. So what this model also does is not only it shows these predictions for different organisms, it also shows these predictions for different cells. So it would give us the prediction in the skin cell, it would give us the prediction in heart, liver, etc. So we get a very dense array, like multidimensional array like structure, which has on one axis these different modalities, and on another axis we have different cells, and we can extract whatever information we need from this for our results. So we talked about the model now, and now let's talk about how the training was done for this. So the training was done in two parts. We have the pre-training step, and then we have the distillation step. So this is a new concept that is being used in a lot of models now. In the pre-training step, they use fourfold cross-validation to generate one model. So what we do is we take 3/4 of the genomic intervals. What genomic intervals means is we have a really long 3 gigabases length DNA, and it's broken down into different intervals. So we take 3/4 of those intervals to do the training on, and then we do the test on the unseen 1/4 of the genomic interval. So that's fold cross validation that is used to generate fold specific models. That's step one. And then we also generate all folds model. So this model is trained on all available intervals. So the 3 gigabases are broken down into multiple intervals. You take all of those and you create the all folds model. So we have these two models. The first one is used to evaluate the generalization, and the second one is used to serve as a teacher for the next stage, which is the distillation process. So we already looked at the student teacher kind of model in alpha genome as well, where you have the original model and then the another model learns from that by adding by introducing some data augmentations and other noise. Here we do a similar thing where you take the original teacher model, you add some random shifts to the DNA. So for example, you have the TC here. If you do a rotation kind of shift, it comes here. So that's one kind of data augmentation you do. And another kind is reverse complementation. So what reverse complementation means is that in DNA, each of these bases are complementary to another base. So G is complementary to C, A is complementary to T, and you take the complements of all of these and then you flip the sequence. That's the reverse complement. And that's important because that is what happens within the cells when DNA changes into RNA. So you also do these reverse complements. That's another kind of data augmentation. And finally, for noise, you add some random mutations. So you change T to G and you change A to C. So these are the changes that we do. After doing these changes, we train the teacher model on these changes, and then we get the student model. And this process is the distillation process. And these are the two steps that we do to get the final model. Now, let's talk about the impact of this model. First is that it's a unifying model. So far, all the AI models that we had in this area, they only focused on a particular modality. So if we go back here, we had this initial model called Informer, which would predict the RNA sequence. So that's one model which did one thing. Then we have this model called Pangolin, which which predicts splice sites and splice junctions and all of these splicing related things. That's another model. So we had these different models, and each of them did a different thing. But alpha genome came along, and they were able to do all predictions in one go and unified the whole process. So that's amazing. The next thing they did is long range context, which is, like I mentioned, the older models operated on much smaller lengths of DNA, but this one was able to take 1 MB of DNA at a time and in one go do the predictions. So how that helps is, it helps us understand these interactions between different parts of DNA, which is not possible if you're looking at only small parts, because the interacting regions of DNA could be much further along. So the longer the context, the better in this case. The next best thing is base level precision, because this model takes us input the DNA sequence and looks at these uh, individual positions having A, G, C, and T at base level. And finally, currently it's the state of the art, as it has better performance in many of these genomic modalities. So for example, I mentioned that Informer and Borzoi do RNA sequence prediction, but alpha genome actually outperforms it in this area, even though it's doing that along with all of these others. There are some areas where it is comparable to the existing state of the art model, but in many areas it's actually exceeding them. So being able to do that along with having a unified model is a great thing. What are the applications of this model in when it comes to bioinformatics? And I'll particularly like to focus on the biomedical area because that's my area of interest. So first, it helps us improve the understanding of different diseases. So I mentioned that we have different variations within the DNA among ourselves. So some of these variations are just for these basic changes that we see in our eye color and skin color, those kinds of things. But some of the changes also lead to a higher risk of diseases. So if you're able to predict what each of these variations do, we can we can better pinpoint what the causes of these diseases are, and that would help us in drug discovery, in better determining what medicines to administer to a particular patient, and so on. This also forms the basis of this field called precision medicine, where the medicine you get depends on your genomic makeup. So that really helps in the efficacy of a medicine, because like I mentioned, how one person responds to it varies from how another person responds, and one of the factors that cause this change is actually our DNA. So depending on which variations we have, we might respond differently to different diseases, medicines. So that's one. The next thing is synthetic biology. So with because this is a LLM kind of a model, we are also able to design new synthetic DNA with cell type specific regulatory functions. So as I mentioned before, this also gives the predictions for each cell type, like it gives the predictions for skin, how the variation would take effect in heart, same functions in different cells and performing those same regulatory functions. Finally, this helps us in accelerating fundamental research. So aside from any direct impact, just being able to understand the different variations will help us better understand the molecular other areas of research as well. So we have looked at two of the major AI models in this area now. And now I would like to talk about what the road ahead looks like in this field, what the future direction would be, what the challenges are. Is the timeline of how these models evolved, especially like at least the ones that gained a lot of recognition. We of course have in 2018 and 2025, but I'm focusing in the more attention and the ones we most commonly use in research. So the first one that gained tremendous attention was AlphaFold, as mentioned, because it solved this long-standing challenge and went on to win the Nobel Prize. Then came Informer. This was the first one to model long range dependencies in DNA regulatory code. Of course, this was not as long range as alpha genome, but it still was a big leap from what we had before. So a key model in this area. And then Borzoi came along, and it just led to improvement in speed, improvement in interpretability. That was another leap. Evo is a little different. Um, Evo is a little different. It brought LLM scale training to genome interpretation. So this was one of the first models to introduce that. I think this was introduced by the ARC Institute. And towards the end of this presentation, I've also added a slide with references in case you want to learn more about any of these. And finally, now we have Alpha Genome, which integrates different modalities and interprets the unseen and unknown parts of the DNA, which we refer to as the dark matter. Now, what are the challenges in this field when it comes to applying machine learning in bioinformatics, but also specifically in the biomedical domain? The first is data quality and bias. So as we know, the models learn from whatever data we give them, and if the data is of poor quality, the model is going to be of poor quality. But something even more important when it comes to biological data is the aspect of bias. So if there is any sort of bias encoded in personal information, that's also going to translate into the model. So for example, until recently, most of these huge public data sets or even protected data sets that we had for biological research were based on data sets obtained from Europe. So that's obtained from people of white ethnicity, white population, Caucasians, and that has very little data from people from Indian ethnicities. So any kinds of predictions that this model makes, it's going to be more accurate for that population, but not for our population. So that's how bias can lead to decrease in performance. But this is something we don't find out about because we train it on the data that we have, which is mostly European data, and we also test it on that data. And in theory, it looks accurate, but when you want to
Yes. Sorry, Akshada, and it looks like you had a temporary loss of connection.
yeah,
And okay,
Okay.
are you able to see my screen now?
no, the presentation is not there.
Okay, sorry, give me a minute. What about now? Is it
Yes.
okay?
Yes.
Okay. Yeah, I'm not sure where I left off, but I'm going to start from here hopefully. So I'll go back a little bit. I was talking about the challenges, and I had covered the data quality and bias part, which I hope you were able to hear. And next, I was going to talk about the high dimensionality with low sample size problem. So why do we have this problem? As mentioned before, all of these models are actually trained on experimental data that we get from all of these different experiments. And each of these individual experiments are very time consuming. So if you want to look at the variation from one like one variation in a DNA sequence in a single person, you have to do all of these different experiments which take days. So even collecting like a single data point can take days. And in this case, you can imagine that we would end up with very low sample sizes. So that's a problem because these huge models need huge data sets. And another problem is high dimensionality, because in biology, you have to look at how a particular variation affects different aspects of molecular biology. And this ends up in like a large complex data structure that you have to deal with. And that also ties into this heterogeneous and multimodal data. So in the current example, we were only looking at sequences, but there are also image data that we can incorporate into these models. There are also structural data. So all of these different kinds of data have different data structures. So they're heterogeneous, and we need models that are able to handle this different kind of data structures. Another important aspect is interpretability. So for any model to be important in a biological context, you have to be able to interpret it. If you just give an input, get the output, and treat the model like a black box, it's not going to be very reliable, especially in a clinical setting, because lives are at stake there. So you need to be able to tell how the model actually predicted a particular outcome. There needs to be some way to interpret its reasoning and understanding of how it came up with the result, and that will help us get more assurance and more confidence in the output that it's predicting. So interpretability is very important, especially in a biomedical setting. Next thing is reproducibility and generalization. So for example, if we talk about the example I just gave a while ago, which is models are trained on one population, but then they are also applied on another population. So if you have a model which was trained on predominantly white population from Europe, it is not going to be very generalizable if you try to apply that in a clinical setting in India. So that's an important challenge to tackle, to have diversity in the kind of data we use. And also reproducibility is important here. Finally, there is this translational gap in clinical adoption. It's mainly because of all of these different challenges. There are worries about bias getting incorporated in data, and there is worry about interpretability. And because of this, even though we have a lot of amazing models, it's not applied in a clinical setting, and we need to go a long way before it finally gets adopted in a clinical setting. And to do that, we have to mainly solve the problem of interpretability and bias. Finally, if it does go to a clinical setting, another important thing to deal with is data sharing, privacy, and ethics. So healthcare data, which is the kind of data we use in biological models, is very sensitive, and there need to be a lot of rail guards to keep it safe. So what happens is that also slows down the research process, because if you want to do a new project, you have to go through a lot of administrative process to get access to that particular data. This is a problem that I'm also currently facing as I'm trying to get access to a huge data bank, but there are compliance issues which make the process very. So we have to. I think there's some noise. Can Okay, thank you. Yeah, so data sharing is an important part to deal with. You have to make data sharing easy to speed up research, but at the same time, ensure that the privacy of the participants who share their data is not compromised. So privacy and data sharing come hand in hand. And then there's also the challenge of ethics. In general, this is a challenge in AI, but it's a bigger challenge when it comes to biomedical data, just due to its sensitive nature. So to summarize everything that we have seen so far, machine learning enables insights from complex high-dimensional biological data. This is not something that's possible through traditional data science, like using statistics and general analysis. So machine learning really helps in solving these intricacies. And the applications of machine learning in bioinformatics span genomics, which is studying the DNA sequence, proteomics, which is studying the proteins, clinical informatics, which is understanding like it affects its effects in a clinical setting, in healthcare, medicines, etc., and then drug discovery. Some of the key advances that I covered in this presentation are AlphaFold, which was a model that revolutionized protein structure prediction, and AlphaGenome, which did functional effect prediction for variants. Uh, I also touched on some of these other models like Borzoi, Informer, or Evo, but we have not gone into the details. But if you're interested, there is a summary slide. There's a slide at the end with the links for it. And currently, the big opportunities for ML in bioinformatics are that it has led to accelerated discovery. So since many of these results can be obtained without experiment now, it can speed up biological research in general. It can also accelerate precision medicine. So I talked about precision medicine a while ago. It is when uh, we can be administered different medicines based on our genetics, and that would improve how we respond to a particular medicine, and that would in general improve the course of the disease. So that's another big opportunity for applying machine learning in this area. And finally, in general, it will help us get a deeper understanding of biology and its applications. It can be wide across these different subfields and maybe even outside biology in general. And finally, we also talked about the challenges that lie ahead. And some of the key challenges, according to me, are interpretability, privacy, integrating multimodal data such as images, like clinical X-ray images, for example, the text data, numerical data, and then in the end, finally, being able to translate all of these applications in a clinical setting, because that's when we'll be able to really use it in a day-to-day context. So I think this would be equivalent to how now in terms of, in case of general LLMs, we have come to the point where all of us can use ChatGPT on our laptop and we can rely on it for a lot of different things. So when a lot of these challenges will be solved, we'll finally be able to see AI being used in a healthcare setting as well, to do predictions, to do personalized medicine treatments, etc. And these are references to all of the models that I've talked about. Most of these are links to blogs by the companies that have developed this model. But Informer and Borzoi does not have a blog, but they have a paper where they talk about these models in details. So thank you everyone for listening. And I think, yeah, we have 10 minutes if there are any questions or just to take a break. Thank you. Are there any questions?
Participants, it's a very good time to interact with Akshatha. She has been a leading researcher in this area, and she will be quite happy to give her insights in future developments, and future research in the area of bioinformatics.
Participants, if you have any questions, you may ask.
Uh, Otherwise, I think, yeah, there are questions, I think. Akshatha, can you see the chat window?
Yeah, so Anjali asks if there are any free data sets available. Yes, so for for a lot of different kinds of things, we have free data sets. If we are talking particularly about clinical data, uh, those are not free, as in not in terms of money, but more about access. So if you want to do any kind of clinical research, there might be a small subset which is freely available, but to get a large enough subset to do machine learning kind of work, you will need to write up a proposal on what kind of research you're trying to do, submit this proposal to regulatory organizations who will evaluate it, and then just give you access to limited parts of the data that are necessary for your work. This is for clinical data, but then otherwise, if you talk about other areas of biology like general DNA data, protein structures, all of those are freely available. So yeah, a lot of biological data is available on the website EMBL-EBI. I can quickly also share the link if you give me a minute. So I've pasted the link here to the website for EBI. They host all of the public data sets that are available for different areas. Um, okay, someone has asked about organization details. I can share my current UT Austin email ID. Give me a minute.
In that case, I think we can conclude. We all know that bioinformatics is an area that is explored, that is currently being explored, and that's again an area where a lot of innovations are happening. And it is nice to see that Akshatha Nayak has come back to her alma mater to help us to organize this faculty development program. And to summarize the session that she has taken, she started with the applications of bioinformatics and explained how AI can be of help in these areas. And then explained the challenge of protein folding, and she then continued with the predictions from DNA and described the alpha genome model. And she concluded with the future directions of research in the area of bioinformatics. And from the side of Department of Computer Science and Engineering, Rajiv Gandhi Institute of Technology, I would like to extend our sincere thanks to our proud alumnus, Akshatha Nayak. Thank you once again.
Yeah, thank you all for listening, and I was very happy to share my insights on this. Thank you.
Thank you very much, Akshata, for coming here as a resource person. As I told during the beginning, we are really proud and happy to have you as a resource person for our faculty development program. You were really a role model while studying. You will continue doing that. We are sure about it. For a person to be a successful engineer, he or she should have engineering knowledge, then academic scores, skills, and attitude. Not only that, Akshata had all these skills in abundance, she motivated others in the class also to have all these skills. That made you distinct from other students. And sincerely, we wish that you will reach more heights of success in your life in years to come. Thank you. Thank you once again, Akshata.
Thank you so much for your kind words and wishes. Thank you, everyone.
So the resource person for the next session has already joined the Google Meet. I think we can take a break for 5 minutes and we will start the session sharp at 7:30. Thank you.